over the world, high-grade ores are in increasingly short supply, and this is why poorer ore bodies must be exploited. Microbial leaching is one possible means of recovering metals from low-grade ore bodies, worked-out mines, and different waste materials which are uneconomic to utilize with conventional methods. The microbial leaching process offers considerable advantages over conventional techniques: lower energy requirements, lower capital costs, and in addition, it is more acceptable to environmentalists. Metal leaching has a long history, although the role of bacteria has only recently been realized. The presence of copper in acid mine drainage waters was observed by the Phoenicians and Romans, and the latter probably recovered the metal from this source. The earliest large-scale leaching operation for copper was carried out at Rio Tinto in Spain in the mid-18th century, but it was not until the 1960s that the involvement of bacteria in this particular system was confirmed. Similar natural leaching has been observed in many areas of the world for a long time. These acid drainage waters have caused severe pollution problems in surrounding soils and waterways. Bacteria of the genus Thiobacillus, which oxidize sulfur compounds, and of the genus Metallogenium, which oxidize iron, are to be found in the early stages of naturally occurring leaching processes. They have optimum pH values of between pH 4 and pH 7. The importance of this group of microbes for the leaching process may be that they lower the pH. The actual ore-leaching organisms are the acidophilic bacteria, which are capable of oxidizing iron, sulfur, or different sulfides. This group contains, for example, the vibroid Leptospirillum ferrooxidans, Thiobacillus thiooxidans, and Thiobacillus ferrooxidans. Thiobacillus ferrooxidans is the most studied and most important representative of this group of bacteria. It was first isolated in 1947 from acid mine drainage of a coal mine. Thiobacillus ferrooxidans is a small gram-negative rod-shaped bacterium with one or several flagella. It is a chemolithotrophic organism, which means that it grows in a totally inorganic environment, using ferrous iron, elemental sulfur, or sulfur compounds as a source of energy. Carbon dioxide is its principal carbon source. Atmospheric oxygen is the principal electron acceptor in the oxidation processes. In addition, Thiobacillus ferrooxidans needs only a few inorganic salts for growth. There is evidence that some strains of this organism are able to fix nitrogen. The optimum pH for growth is between 2 and 3.5, and the optimum temperature is between 20 and 35 degrees centigrade. As can be realized, Thiobacillus ferrooxidans is a very suitable organism in acid leaching processes. It is also very versatile in attacking metal sulfides and has been reported to be able to oxidize several minerals. In addition to this versatility, Thiobacillus ferrooxidans is able to develop a high degree of tolerance to metal ions in solution. The concentrations of various metal ions that it has been shown to tolerate are considerably higher than the inhibitory levels associated with other groups of organisms. This undoubtedly contributes to its success in oxidizing a wide range of complex ores from which these ions can be extracted. However, the growth of the organism is inhibited by low concentrations of mercury, silver, and molybdenum. Thermophilic bacteria capable of oxidizing iron, sulfur and some sulfide minerals have been isolated from actively leaching dumps. In addition, many heterotrophic bacteria, fungi, algae and protozoa have been found in association with leaching operations. The role of these organism groups in the ecology of the leaching process is still obscure. Two mechanisms of microbial ore leaching have been proposed: direct microbial attack on mineral sulfides and indirect attack via ferric iron and sulfuric acid, which can be generated by microbial oxidation of soluble ferrous iron, iron sulfides and elemental sulfur. It is evident that both mechanisms contribute to the leaching process, but in nature it is impossible to determine to what extent the mechanisms apply. Ferrous iron produced in these examples of indirect microbial attack with ferric iron on different minerals can subsequently be reoxidized by iron oxidizing bacteria, setting up a cyclic reaction. Sulfur produced in the reactions can be oxidized to sulfuric acid by bacteria. The microbial oxidation of ferrous iron is 100,000 times faster than chemical oxidation. The mechanisms of direct oxidation are not yet completely understood, but experiments with minerals containing no iron have led to the postulation of direct microbial attack. Scanning electron micrographs have revealed numerous bacteria attached to the surfaces of sulfide minerals. In ore suspensions, the majority of the organisms are attached to solid surfaces, although it has been shown that attachment is not essential for the occurrence of microbial leaching. Economically, the most important metals that are presently recovered by microbial leaching are copper and uranium. Many countries, including some developing countries, commercially apply microbial leaching mainly for copper and uranium recovery. An important basis for the successful commercial application of microbial leaching is the laboratory experiments that are carried out in laboratories throughout the world. The first task is to find suitable organisms. In the isolation of Thiobacillus ferrooxidans, a solid medium containing ferrous iron is used. Silica gel, or pure agar at low concentration, can be used as a solidifying agent in the growth medium. The colonies are seen on the membrane filters after 2 or 3 weeks of incubation as brown spots about 1 millimeter in diameter. The brown color is the color of precipitated ferric iron. The growth of the organism in ferrous sulfate liquid medium is indicated by the change in the color of the medium from green to brownish, which is the color of ferric iron. Different shades of the brown color can develop depending on the ferric iron concentration and on the pH of the medium. If the pH is around 1.8 to 2 or more, ferric iron will be precipitated out. The growth of the organism on ferrous iron can be monitored by potassium permanganate titration of the residual ferrous iron in the medium. The number of Thiobacillus ferrooxidans cells can be counted by the most probable number method, using 5 replicate tubes of each 10-fold dilution. The medium used is the ferrous sulfate medium. After 3 weeks of stationary incubation, the amount of positive brown tubes in each 5-tube series is checked and the numbers of bacteria are determined. If sulfide oxidizers are grown on iron sulfide, the black sulfide powder will disappear and the pH of the medium will decrease while the ferric iron and sulfuric acid concentration of the medium increases. Laboratory scale leaching experiments are mainly made to check the amenability of the particular ore sample to microbial leaching and to find the optimum leaching conditions. Some of the factors studied are the composition of the leaching medium, pH, reduction oxidation potential, temperature, particle size of the ore, and various leaching organisms. The experiments are commonly made using shake flasks and different percolation techniques. Conical flasks can be used in shake flask experiments. The liquid medium, containing the finely ground ore, can be sterilized before the inoculum is added. The leaching medium consists of the mineral salts and the ore sample in a dilute sulfuric acid medium. The pH is about 2. The flasks are incubated in a shaker for a couple of weeks. During the incubation, evaporation losses are replaced with water and the pH is adjusted as necessary. Samples for metal analysis, pH and electrode potential measurements and total bacterial cell counting are taken at certain time intervals. The total number of the bacteria in the liquid phase can be counted using a counting chamber. The metals are analyzed using an atomic absorption spectrophotometer. The plotted leaching curves show the progress of the microbial leaching process. Metal sulfides, their textures and interrelationships, are conventionally examined by means of ore microscopy, which is based on the high reflectivity of sulfide minerals. Partly leached samples, or leaching residues, are washed, dried, mounted in plastic and polished. Much information about the dissolution process can be obtained when textural changes in the mineral grains are examined. In stirred reactors, the leaching process can be monitored under controlled conditions. Microbial leaching can also be tested using percolation leaching techniques. In this method, The solvent containing the bacteria is circulated through the ore bed, in a manner similar to the commercially used leaching methods. The circulation of the liquid can be established by airlift or using a pump, and in the different percolator models, the solvent can either totally cover the ore bed or only wet the ore bed drop by drop. The ore material used in the percolation techniques is more coarsely ground than in the shake flask experiments. The experiment is run for months. Samples are taken to monitor the leaching process. The growth of the bacteria can easily be seen from the change in colour of the liquid. Larger scale leaching experiments can be made in big columns using the percolation technique. These columns may be several metres high and may contain up to 2 tons of ore material. The predominant methods used for commercial applications of microbial leaching are dump leaching, heap leaching and in situ leaching. Dump leaching is used to extract mainly copper from low-grade oxide and sulfide minerals and waste materials removed from open pit mining operations. The copper content of the ore material varies between 0.1 and 0.4%. The height of the dumps usually varies between 15 and 200 metres. The amount of ore material may be from some thousands to hundreds of thousands of tonnes, including large boulders weighing many tonnes, as well as material of fine particle size. Leach solutions are usually introduced on the top surfaces of the dumps by flooding or sprinkling. The solutions then percolate through the dumps, where the ever-present bacteria operate by oxidizing sulfide minerals and ferrous iron. The percolating liquids are collected at the base of the dumps. Copper is recovered from the pregnant liquid by cementation with scrap iron, electro-winning, or by solvent extraction. The barren solution is then returned to the dumped surfaces for reapplication. In some systems, the reoxidation of the iron in the barren solution is allowed to take place in oxidation ponds before reapplication. The leaching cycle in dumps is carried out for years. Heap leaching works in a similar way, but it is smaller in scale and uses finer and more concentrated ore. The third commercially important method, underground in situ leaching, is done especially in old, abandoned uranium mines. The stopes are flooded or hosed to encourage microbial action, and the pregnant solution is collected in tunnels at the lower levels and pumped out of the mine for extraction of uranium. For the treatment of low-grade ore bodies, for which the cost of conventional mining would be prohibitive, the ore body may be leached in situ by pumping the leaching solution into drill holes and allowing the liquid to percolate to a lower collecting area from which it can be brought to the surface. The permeability of some rocks can be improved by fracturing them with explosives. Biohydrometallurgical processes need not necessarily be limited to low-grade ores. There are indications that after optimization of the biological and technical process parameters, ore concentrates can also be treated economically by biohydrometallurgical methods, which do not contribute to air pollution. Advances in microbial leaching may also allow the exploitation of ores that, although of reasonable grade, cannot be treated conventionally. The microbial leaching of metals from different industrial waste materials has also given promising results on a laboratory scale. Removal of pyrite from high sulfur coal is also one application field for acid microbial leaching processes. For environmental reasons, high sulfur coal is not burnt because of the large quantities of sulfur dioxide produced. Laboratory studies have shown that pure or mixed cultures of thiobacilli can rapidly and effectively remove pyrite from such coal, producing a clean product. Apart from acid microbial leaching can also be applied using heterotrophic bacteria like Bacillus circulans and fungi like Aspergillus niger. These microbes grow at a higher pH and produce organic acids which can be used to extract, for example, iron from sands and aluminium from silica. Finally, it is worth mentioning genetic engineering in acidophiles. However, our lack of knowledge of the genetic material of the thiobacilli prevents advanced studies in this particular area. With genetic manipulation, it would be possible to improve the tolerance of the thiobacilli to some toxic materials in the leaching process on the one hand, and the leaching activity towards different sulfides on the other. Considering the diminishing natural resources of the world, the growing demand for metallic raw materials, and the advantages of the biohydrometallurgical processes, more effort should be put into research and development in this area. The versatility of the organisms used in microbial leaching processes offers great opportunities for the worldwide application of this biotechnical method.